Hi everybody, and welcome to Biology 3270-5270 Microbial Ecosystems. This is my first recorded lecture for this course. As I hope you all read in my email to the class, I'm going to try to record my lectures and post them on Canvas. You guys will watch these videos and then we'll discuss them and answer Canvas quiz questions about them in class. So the whole course format that I came up with sort of at the last minute that I hope you guys like kind of depends on you watching these videos. So I really appreciate you taking the time to watch them. And I will try to be as concise as possible. I'll try not to talk very fast, but I will try to only say things with as many words as they require. And then I can elaborate in class. And so I'll try to use few words when giving these lectures and these videos. All right, let's do this. So this course is called Microbial Ecosystems, and so that first word, microbial, includes all life on the planet that is microscopic. That is, that you need a microscope to see it. That's really the definition of microbe and microorganism. Sometimes I see it that it's defined as single-celled organism, and that also works. I kind of like just microscopic because some of these organisms are not necessarily always living as individual single cells. So I just like microscopic. And that includes all three domains of life. So archaea and bacteria, you probably know, are microbes. And also most eukaryotic organisms on Earth are also microscopic and are microbes. So the red branches on this tree of life, which is just one version of the tree of life that you could draw, but it's one valid version. These three red branches are basically the only branches on the whole tree of life that do not include any microbes. These are the big multicellular organisms. There are some other kinds of algae and other organisms that are also multicellular, but the plants and fungi and animals, those are our main multicellular lineages. And all these others, these are all single-celled microscopic organisms. So even the eukaryotic domain is full of microbes. In short, most of life on the planet is microbial. So we're going to be talking about the ecosystems on Earth that make up most of the life on the planet. And much of the modern research that we're going to talk about in this class, including the papers that we will discuss and the papers that you will read and you'll write about, make use of this revolution in technology that made the modern field of microbial ecology possible. That is the technology of DNA and RNA sequencing. This revolution in the 90s going into the 2000s completely revolutionized the field of microbiology and our appreciation and knowledge of microbial diversity. And in this case, you can kind of trace this revolution back to a single scientist. Normally, historians of science caution that attributing a discovery or invention to a single person is almost always a simplification. And to some degree, this is also a simplification, but Carl Woese was really single-handedly responsible for much of the revolution in microbial diversity that we sometimes take for granted today. So who was Carl Woese? He was a physicist who decided that he could use quantitative techniques that he borrowed from physics and math to understand biological diversity. That was his goal. He was uncomfortable with the kind of squishy ways that biologists were using to categorize diversity on Earth at that time. You know, things like measuring the lengths of bones and doing comparative anatomy and saying this thing must have evolved from that thing because, explanation. And he wanted to use math to make a tree of life. And he, to do that, he knew that he needed to use molecular sequences, that is DNA and RNA and protein sequences. And the technology for doing that was just becoming available in the 70s when he started doing this. And with some thought and trial and error, he decided to use the ribosome, and specifically ribosomal RNA, as his marker to measure the diversity of all life on Earth. He chose the ribosome because all organisms on the planet have a ribosome, so that is good if you want to measure the diversity of all life on the planet. And he knew that he could sequence the gene that encodes the small and large subunits of the ribosomal RNA. And even at that time, they were starting to appreciate that it's the RNA portion of the ribosome that really does all the work, and the proteins are kind of accessories. So for a lot of reasons, this really seemed like a good idea. It's universal, present in all life. He can sequence just 1 or 2 genes that encode the small and large subunits, so that would be practical in those early days of DNA and RNA sequencing. And also there was already an appreciation that this thing that the ribosome does, which is make proteins from RNA, must be ancient. Like that must have been one of the very first biochemical functions that was present on Earth that kind of made possible the rest of life. So there are lots of reasons for him to choose this. He made an early version of the 3-domain tree of life in this in 1977. And then a figure that is more well known now is in this paper from 1990, which was kind of after he had spent 13 or more years refining his technique. And this 1990 paper, this is figure 1 from that paper, was his kind of grand official proposal that all life can be characterized into 3 domains. When I was in high school, I learned that there were 5 kingdoms: plants, animals, fungi, protists, and Monera, which includes both bacteria and archaea. I also learned that there was this other idea of 3 domains when I was in high school, but I kind of learned both the 5 kingdoms and 3 domains. And now we have completely adopted Carl Bose's proposal that all life really belongs to 3 domains. Every single organism on the planet is either in the bacterial, archaeal, or eukaryotic domain. Okay, so that was a huge biological discovery, and he made it with this new technique of using DNA and RNA sequences to make trees. So it was not only a major just conceptual discovery that all life belongs to 3 domains, but it was also a radical technical innovation. This is the first time anybody had made any kind of tree at all with molecular sequences, and he did it to make this huge discovery. So it was really a remarkable episode in the history of science. And that started the field of microbial diversity, or environmental microbiology, or microbial ecology, however you want to call it, that is extremely productive and one of the hottest fields in science right now. So after that bit of history, I thought I would start giving you some examples from the modern day. We can talk more about Carl Bose perhaps in class. So examples of archaea. What are archaea? Because for a long time, basically until Carl Bose, we didn't realize that they were different from bacteria. There's a fantastic example of archaea very close to home, which are the halophiles, that is salt-loving archaea in the Great Salt Lake. And they are one of the few organisms that can survive in the north arm of the Great Salt Lake. So if you've ever driven all the way up to the north and looked, you'll see that the water is red, which is pretty weird. It's one of the weirdest places I've ever been, the north arm of the Salt Lake. It's red because of the archaea. You may have heard that the red color is from algae. That's just plain not true. I don't know where that came from. It's red because of archaea. So we actually can see archaea from a plane. If you're flying over it in an airplane, or actually from space, you can see the red color of the archaea that are super abundant in the north arm of the Salt Lake, and that causes the red color. It's red because they have pigments that they use to do photosynthesis. Other examples of archaea are methanogens. Methanogens are archaea that make methane. They are the only organisms on the planet that can make methane. So all of the methane on the planet basically comes from this one group of archaea. Methane is predominantly of biological origin. There are non-biological origins of methane as well, but it's mostly from archaea. I should actually say that there's recent research in the past year or two that may be additional sources of methane that are not from methanogens. But that was a brand new discovery, and it was remarkable because before then it was thought that archaea were in fact the only sources of methane on the planet. So the methanogens live in places like your own digestive system. So the gas that you emit after eating beans, that's from archaea living in your digestive system. Cows have a lot of methanogens living in their digestive systems. Swamp gas is methane from archaea. Rice paddies, same thing. Sewage systems, it's all archaeal methanogens. Here's a cool picture of methane that was trapped underneath a frozen lake. And here are researchers who are studying melting of permafrost and lakes in the Arctic and how that might lead to the release of methane, which would be really bad news for our climate. And as a photo op, here they are poking a hole in the ice and then lighting on fire the methane that comes out of the hole in the ice, just to show how much methane is being stored underneath these frozen lakes. And that methane was generated by methanogens living in the bottom of the lake. Archaea are often described as extremophiles, and many archaea are extremophiles, but also many bacteria are extremophiles. So I think it's actually incorrect to describe archaea as extremophiles, because there's nothing necessarily more extreme about archaea in general than about bacteria. Both archaea and bacteria include species that are extreme in various ways. So for example, the current record holder for hottest temperature for growth is currently held by an archaeal species, in fact a methanogen, growing at 122 degrees Celsius. That's above the boiling point, but it's growing under pressure, so the water is not actually boiling, because at the pressure is where this organism lives. The boiling point is higher than 122 degrees Celsius, so it's still living in liquid water. The boiling and freezing points of water seem to be one of the main limits for life on Earth. As long as you have liquid water, no matter what the temperature or other conditions of that liquid water are, it seems like you can have life. Again, similarly with the coldest temperature for growth of any species, it's minus 15 degrees Celsius, which is below the freezing point, but it's extremely salty water, so it's actually still liquid water at minus 15, which is how you can have growth. And that is actually a bacterial species. Right? So both bacteria and archaea can be extreme. The most basic, the highest pH for growth is currently held by a bacterial species. The most acidic, currently held by an archaeal species. No, so you get the point. This is just to help you remember what, sorry, I was going to say pH, but this is temperature. These are some examples of temperature limits for kinds of microbes. And you can see that bacteria and archaea are the only ones that can live at super high temperatures. Eukaryotes tend to only be mesophiles, that is, organisms that can only live at what we consider to be normal temperatures. And here's a Yellowstone hot spring and showing the abundant biofilms that these thermophilic bacteria and archaea make associated with these hot springs. Okay, so in a kind of typical microbiology course, we would go into detail in the molecular differences between bacteria and archaea, and especially with the eukarya. And I do want you to appreciate the similarities and differences among these three domains. It's important for your general biological education to know something about why we think these three domains are distinct. It's not only because of Carl Woese's tree. His tree was the initial discovery that led to many specific predictions that there should be distinct differences among these three domains. And then after he made those predictions, tons and tons and tons of research tested those predictions, because almost every single biologist on the planet was actually extremely skeptical, and many were even angry about Carl Woese's predictions, and they thought he was outside of his lane and he should go back to physics and stay away from biology. And so there were many biologists who were ready to completely disprove Carl Woese and show how he was an idiot, and that is not what happened. In fact, all of the predictions that Carl Woese made, as far as I can tell, maybe there was one prediction that was not did not come through, but I think basically they were all correct. And so this table, there are many versions of this table you can find in various textbooks, is one summary of the differences between the three domains that were predicted by Carl Woese's tree. So, for example, bacteria and archaea usually have circular genomes. The DNA is a circle. Eukaryotes not. So that's one similarity between bacteria and archaea. And there are many similarities between bacteria and archaea, which helps explain why it took us so long to appreciate that they were different. But there were also really important similarities between archaea and eukarya that we'd never realized until Carl Woese predicted it. For example, the initiator tRNA, if you don't know what that means, that's totally okay. It just means that the first codon on every single protein in archaea and eukarya is the same, this methionine, whereas bacteria use a different tRNA to start all of their protein synthesis chains. So again, if you don't know what that means, it's okay. It's just an example of really, really basic biochemistry that is the same in archaea and eukarya, but different in bacteria. What's another example? The archaea also use the Tata box. If you have heard of that in cell biology or a previous biology course, you were learning about it in the context of eukaryotic cells. Archaea also do the same thing, whereas bacteria have something totally different for promoters. Again, if you don't know what a promoter is, it's okay. The important point is that there are several similarities between archaea and eukarya that were predicted by Carl Bose's research. And then there are also some important things that are unique to the archaea. One example is their lipids. So they have ether-linked lipids, that is the chemical bonds and the lipids of their cell membranes are ether bonds, which are not ever found in the bacteria and eukarya, or they're not the main chemical structure of the lipid membranes in bacteria and eukarya. So that's one example of how the archaea are quite unique. And there are many, many other examples. These are just some of the really basic biology reasons why these three domains are distinct. They share some important similarities, but there is no organization that you can make that is simpler than the three domains, nor any other different organization that explains all of the available data. The three-domain structure is extremely well supported now. There's just a huge amount of evidence in support of the three domains, and there is no other system for organizing the diversity of life on Earth right now that enjoys as much support as this one. Here's just another version of that same table that might be easier for you to study. Not that you necessarily need to study for any exams in this course, but there you go. It's just another version of it. Here's yet another version of it, just to show that it's the similar information organized differently. This one has an interesting one, which is that bacteria are the only ones that are sensitive to antibiotics. So luckily, no archaea cause any diseases that we know of. And that's good because they are not sensitive to antibiotics. So if we do ever suffer from any infections from archaea, we would not be able to use antibiotics to fight those infections. Those things are probably not a coincidence. There's probably a deep evolutionary reason why archaea don't infect us, perhaps because their biochemistry is so much more similar to us. And the biochemical differences in the bacteria might explain why they are so often pathogens of us. That's actually, kind of a deep biological mystery. But yeah, basically archaea don't cause disease, and they also are not sensitive to antibiotics. Here's just more basic biology to make sure we're all on the same page. Eukaryotes are the ones that have nuclei in their cells. So here's a traditional eukaryotic cell from a textbook. Here's the nucleus that contains the genome. Usually the genome is composed of multiple linear chromosomes, whereas bacteria and archaea have these usually a single molecule, a single genome, although I just saw a paper yesterday that was proposing that most bacteria and archaea actually have multiple chromosomes in their genome, multiple copies of the genome inside a single cell. So this is a bit simplified, but that's new research. But anyway, you can appreciate how this is quite different way to organize your genome than this. So that's a really big, important difference between eukaryotes and bacteria and archaea, making the eukaryotes quite unique in the way that they store their genome. This was a big evolutionary moment when the eukaryotes started organizing their genomes like this, instead of the ancestral form, most likely, which was this single circular chromosome. Another example, RNA polymerases. So those are the genes that do transcription, that turn DNA genes into RNA, mRNA transcripts. And if you do a phylogeny of the genes that encode this enzyme, then you'll see that the archaeal and the eukaryotic forms of the enzyme are really similar. So here's this silly, simple phylogenetic tree showing that. And also from this biochemical structure diagram, you can appreciate how the archaeal form and the eukaryotic form are structurally very similar, whereas the bacterial RNA polymerase is structurally and therefore functionally quite distinct from the archaeal and eukaryotic forms. And for that reason, people actually study archaeal enzymes. So they grow archaea in the lab and study their enzymes as a way for understanding human biology and human diseases. So that is a very practical consequence of the fact that archaea and eukaryotes share a much more recent evolutionary ancestor than with the bacteria, which seem to have diverged from the archaea and eukarya longer ago. And that was actually true in the very first tree that Carl Woese predicted. He not only predicted that there are 3 domains, but he predicted that the archaea and the eukarya should share a more recent ancestor than with the bacteria. I mentioned this in the table, but here is a diagram, a figure showing more in detail those ether lipids in the archaeal cell membranes compared to the ester bonds and the lipid membranes of the bacteria and archaea. So that's a really basic biochemical difference. And again, all of that is to show some concrete examples of how we can organize all life on the planet into 3 domains and how all 3 domains are mostly microbial. So I've been using the word microbial. That's definitely the term that I'm mostly going to be using. Another word that often comes up, and we will use it a lot, and we will read it a lot, is prokaryotes, which I'm sure you have heard of before. It is often thought of as the opposite of eukaryote, and we are using the word eukaryote. That is the name of one of the 3 domains, the eukarya or the eukaryotes. And so prokaryote is often used as the opposite of eukaryote, and that would include both bacteria and archaea. But we just talked about how there are a lot of ways that the archaea and bacteria are different from each other, and the archaea are in fact more similar to the eukaryotes than they are to bacteria in a lot of ways. Most of the genes in archaeal cells are more similar to my genes than they are to any bacterial genes. Right? So it's a little bit weird to use this term prokaryote to lump together 2 things that are very different from each other. It would probably make more evolutionary sense to have one term that includes both the archaea and the eukaryotes and excludes the bacteria, but we don't actually have that word. So some microbiologists have somewhat radically proposed that we banish the word prokaryote, that we just stop using the word prokaryote, that it doesn't really have the meaning that it once did. And that, I think, is certainly true. The word prokaryote does not have the meaning that it once did, because it also implied that the prokaryotes were the ancestor. And in that way, it could still have some value. So that is the other viewpoint on the word prokaryote, is that there is some value in pointing out that the eukaryotes are unique. They're unique in having mitochondria and having a nucleus and having a cytoskeleton, which is all this kind of complicated structural parts of the cell that organize it, that the archaea and bacteria don't have. Although I would point out that archaea and bacteria do have cytoskeletons, they just have different cytoskeletons. But fair enough that the eukaryotic one is different. They have lots of compartments like vacuoles and other stuff. They have endocytosis, which is the ability to bring stuff into the cell membrane that the archaea and bacteria don't have, although the archaea might have actually ancestral versions of that. So there's some value in using some kind of word to say that the cellular organization of archaea and bacteria might be more similar to each other than to the eukaryotes. I think that's fair enough, but I also think that there's been so little research on the cell organization of archaea in particular that we really don't know that the archaeal cell organization is similar to the one of bacteria. We just know that they don't really have a nucleus and they don't have mitochondria, but I think that's pretty weak. I think we can all agree that prokaryote is not monophyletic. So if you've taken a biology course before, you know that a monophyletic group is a group that includes all descendants of a common ancestor. And the prokaryotes, the archaea and bacteria, are not monophyletic, right? The archaea and eukarya are monophyletic. They share a common ancestor. But the common ancestor of archaea and bacteria also includes the eukaryotes. So the prokaryotes are not monophyletic. And so we can certainly say that the word prokaryote is not a valid evolutionary grouping because it is not monophyletic. So that's my conclusion that tries to have it both ways, to see points on both sides of this debate of the word prokaryote, that it's a cell biology term that kind of describes cellular characteristics, but it's not an evolutionary term. It does not describe evolutionary past, and it does not describe a monophyletic group. It is not a way to categorize diversity. It could be a way to talk about the ancestor of the eukaryotes, though. That is what the word originally intended. That's why it's called prokaryotes. It's the before the karyotes. That's what came before eukaryotes. And in that sense, I think the word could still be used. It's almost never, that's almost never how the word is being used. But if we wanted to make a change, we could try to decide we're only going to use it in this context, which is, what is the hypothetical ancestor? Because we don't actually know what the ancestor was. What did this ancestor of the eukaryotes look like? What did the ancestor that did not have a nucleus, did not have many mitochondria, did not have any organelles look like that gave rise to the eukaryotes that do? One proposal is that it was in fact an archaeal species, and that's why there are so many similarities between the archaea and the eukaryotes, is that the prokaryotic ancestor was in fact a particular archaeal species. So the prokaryote was this archaeal species that gave rise to the eukaryotes. So that is one proposal. There are other proposals for the prokaryotic ancestor. Other proposals involve it being a bacterial ancestor. I think that is not as popular these days. Another proposal is that it was just an ancient eukaryote, that it has always been distinct from the archaea. And that's an idea that we can talk about in more detail in another class. For now, I just want to say that that word prokaryote could have an evolutionary meaning in that very specific way, not to group bacteria and archaea together, but to describe one hypothetical ancestor of the eukaryotes. Okay, I want to end this lecture by giving you some recommendations for more reading if you want to read more about this huge discovery of the three domains. And one episode that I really enjoy was this fight between Carl Woese and Ernst Mayr, who was a traditional biologist. He is one of the people who authored the biological species concept that you probably learned in a previous biology class about how we currently think about species. And he was very skeptical of the whole molecular sequence revolution because he did not think that gene sequences on their own were going to tell us much about evolution, that there was so much else about biology that you needed to study in order to understand evolution than just some gene sequences. He seems very outdated nowadays because gene sequences have been so incredibly useful, but if you can get past the kind of antiquated nature of his arguments, he actually makes some really interesting points, especially philosophical points about the nature of biology. In this particular example, though, he comes out looking pretty badly because he was quite wrong about Carl Woese's discovery, but it's still enjoyable to read this exchange. So Mayr wrote in 1998, here it must be remembered that Woese was not trained as a biologist and quite naturally does not have an extensive familiarity with the principles of classification. So this is Ernst Mayr criticizing Carl Woese's techniques of phylogeny because he was not following the techniques of making phylogenetic trees that biology students had been trained in for 100 years before Carl Woese came along. And he's doing it differently, and Ernst Mayr is grumpy about it. So Woese wrote a lengthy reply in a long article that he published subsequently, where he said, the disagreement between Dr. Mayr and myself is not actually about classification. It concerns the nature of biology itself. Dr. Mayr's biology reflects the last billion years of evolution. Mine, the first 3 billion. That is, Dr. Mayr is really only talking about animals and plants, whereas Carl Woese is talking about microbes, and microbes were the only organisms on the planet for 3 billion years, and the animals and plants came much more recently. So that's an interesting dig from Woese. He says his biology is centered on multicellular organisms and their evolution. Mine on the universal ancestor and its immediate descendants. His is the biology of visual experience, of direct observation. Mine cannot be directly seen or touched. It is the biology of molecules, of genes, and their inferred histories. Evolution for Dr. Mayr is an affair of phenotypes. That's a quote from Dr. Mayr. For me, evolution is primarily the evolutionary process, not its outcomes. So Carl Woese is interested in how evolution works, not just looking at fossils and skeletons of organisms that are alive today, which is how Mayr worked. He finishes by saying, the science of biology is very different from these two perspectives, and its future even more so. So he finishes with a very open outward look about how biology is going to continue to evolve and change even beyond what these two scientists currently imagine. Here's a link to Norm Pace, who was a descendant, intellectual descendant of Carl Bose. He worked with Carl Bose early in his career. Here is his proposal to eliminate the word prokaryote, which I think is really interesting if you are interested in this. And also here's a YouTube video of him talking about not only the word prokaryote, but also it's a really good history of Carl Bose and the discovery. And he has a much more firsthand account of these discoveries than I do, for example. So I recommend that if you want to learn more. Okay, that is our first lecture. Thank you for listening and watching it, and I look forward to discussing it with you in class. See you then.